So, have you ever wondered what makes some bacterial infections so incredibly dangerous? We're not just talking about the germs themselves, but about this powerful, kind of secret weapon they carry. All right, let's dive in and unmask one of the most potent molecules in the entire microbial world. I mean, we've all heard of E. coli, right? For the most part, it's a common bug. But then you hear about these certain strains that cause horrific food poisoning and even life-threatening infections. So what's the deal? What gives these tiny little organisms such a nasty punch? Well, the answer is actually hidden in plain sight, embedded right on their outer wall. It's like a suit of molecular armor that also happens to be a toxin. It's called lipopolysaccharide, or just LPS for short. You might also hear it called an endotoxin, simply because it's part of the bacterium. And this molecule, folks, this is the main character in our story today.
Okay, so to really get a handle on how LPS causes so much chaos, we first need to pop the hood and look at its design. Think of it like we're disassembling a complex piece of enemy tech. We're going to break it down into its three key parts to see exactly how it all works together. So here's the basic blueprint. LPS is made of three distinct sections, all linked up. On the very outside, you've got the O antigen. That's connected to the core oligosaccharide in the middle, which is then jammed into the bacterial membrane by lipid A. Let's take a closer look at each piece, starting from the inside. Let's zoom right in on lipid A, because this is the real business end of the molecule. This is the fatty part that literally anchors the whole thing into the bacterium's outer membrane. But way more important than that, lipid A is the toxic part. It's the bit that our immune system spots and immediately recognizes as a major threat. Okay, next up is the core oligosaccharide. Its job is pretty straightforward, but absolutely crucial. It's the structural bridge, linking that toxic lipid A to the outer part of the molecule. This core is also essential for just holding the bacterium's outer wall together, making it totally vital for its survival.
And finally, we get to the O antigen, the outermost layer. Now, think of this as the bacterium's camouflage. It's this long chain of repeating sugars, and here's the trick. It's incredibly different from one bacterial strain to the next. That variation is exactly why you hear names like E. coli 0157. This constantly changing surface is a brilliant way for the bacteria to hide from our immune system.
So, if the O antigen is such a great disguise, how on earth does our body ever spot the threat? Well, even the best camouflage can't completely hide the danger underneath. Our immune system has evolved this incredibly sensitive molecular tripwire designed specifically to detect LPS. Let's see how that alarm gets pulled. You know, our body's alarm system for this is surprisingly organized. It's like a relay race. When bacteria die and release LPS, a protein called LBP, that's LPS binding protein, swoops in and grabs it. LBP then acts like a delivery service, handing the LPS off to a receptor called CD14. Then CD14 presents it to the final detector, a complex known as TLR4. And the second TLR4 gets activated, bam! The alarm bells start ringing like crazy and the body unleashes a massive immune alert.
But here's the thing. Bacteria are clever. They're always evolving to get around our defenses. This is a fantastic example of that. So on the left, you see E. coli. Its lipid A has 6 fatty acid chains, which our TLR4 receptor picks up loud and clear. But now look at Yersinia pestis. Yep, the bug that causes the plague. It can actually modify its lipid A to have fewer chains. This makes it a much weaker signal for our immune system. It's basically turning down the volume on the alarm, letting it sneak past our defenses.
So our body detects the threat, it sounds the alarm. That's a good thing, right? Well, yes, but only to a point. Because when an infection gets really bad and there's a ton of LPS floating around, that powerful immune response can start to spiral dangerously out of control. This is where our own defenses can become our worst enemy. In the beginning, the signs are exactly what you'd expect. The immune system spots the LPS and releases signals that cause a fever and inflammation in the area. This is the body trying to, cook out the invaders and call for backup. You can think of it as a controlled, localized fire designed to contain the threat. But what happens if the infection spreads and huge amounts of LPS flood the bloodstream? Well, that controlled fire turns into an out-of-control wildfire. The response goes systemic. Inflammation spreads through the whole body, causing blood vessels to widen and become leaky. This leads to a dangerous drop in blood pressure as fluid just pours out into the tissues.
This completely out-of-control, body-wide response actually has a name, a cytokine storm. Cytokines are the chemical messengers of the immune system. So, in a cytokine storm, the body is essentially screaming danger everywhere, all at once. This overwhelming flood of signals causes massive, widespread damage to the body's own tissues and organs.
And the devastating final result of that cytokine storm is septic shock. This is the point where blood pressure craters and vital organs start to fail because they just aren't getting enough blood. It's a full-blown medical emergency where the body's own defense system is tragically causing its own collapse.
And let's be absolutely clear about how serious this is. The body's reaction to LPS is the single biggest cause of death from sepsis caused by what we call gram-negative bacteria. That's a huge category of germs that includes major players like E. coli and Salmonella. It's a massive, massive problem in medicine.
Now I know that all sounds incredibly grim, but here's the good news. Understanding the enemy is always the first step to defeating it, and the knowledge we've gained about LPS isn't just for textbooks. It has led directly to some powerful, practical ways to fight back.
I mean, this knowledge is being used in critical ways every single day. For example, there's a thing called the LAL test, which uses the super-sensitive blood of horseshoe crabs to detect even tiny traces of LPS. It's how we ensure our medicines and medical devices are safe. We've also figured out how to detoxify LPS and use it as an adjuvant, basically a booster shot for vaccines that really kicks the immune system into high gear. And on the cutting edge, scientists are developing drugs that directly block that TLR4 alarm receptor, trying to stop a cytokine storm before it can even get started.
It really all comes down to this core idea. LPS is a true double-edged sword. For the bacterium, it's an essential piece of armor it needs to survive. But for us, the host, that exact same molecule is a powerful trigger for a potentially fatal disease.
And that really brings us to the big final question. We've already seen how bacteria like Yersinia pestis can tweak their LPS to hide from us. It's a constant, non-stop evolutionary arms race. So as these microbes keep sharpening their secret weapons, the real challenge for science is, can we stay one step ahead?